All right, chapter 4 is called the tour of the cell because we're just going to look at parts of the cell. All right, both prokaryotic and eukaryotic cells. All right. All cells have DNA, right? Just like all organisms have DNA. All cells have a outer membrane called the plasma membrane. It's a boundary that sets apart the stuff inside the cell from the external environment. Also, everything between the plasma membrane and, in the case of eukaryotic cells, the nucleus, is called cytoplasm. Cyto means cell. Plasm is going to be stuff, so it's cell stuff. All right. And then all cells have these little have the cytoplasm, but they also have these little structures that allow them to do certain functions for the cell, and these little structures are called organelles. Like an organ, ELLS is miniature, so these are small organs, right, for the cell. Cell size, these are usually microscopic. Cells are. It wasn't until 1665 when Robert Hooke was credited with discovering the cells. Cell size ranged from 0.1 micrometer to several centimeters. In the case of ostrich eggs, bird eggs have to be large because they have all the material that is there for the young to develop. Red blood cells are about 8 micrometers in size, and this allows them to move in and out of capillaries. All right. Most cells range from 1 to 100 micrometers. A meter is equal to 100 centimeters, 1,000 millimeters, 1 million micrometers. So these cells are, like a millionth of a meter in size. They can be, very, very small. And then 100 nanometers, and that's molecular size, right? I'm sorry, 100. A billion nanometers is equal to 1 meter. So that's molecular size scale. The surface area to volume ratio is the reason why cells are so small. The cells have to be small to allow for enough material to keep them going and thriving and doing their function, but they can't be too large because they have to get things into and out of the cell, and if they're too large, then the cell can't efficiently get things out of the cell that they need to, or bring stuff in to the cell that they need, right? So this, if you blow up a balloon, the skin of the balloon, the surface area, it increases, right? But the volume inside of the balloon is actually expanding at a greater rate than the surface area. And so in order for cells to efficiently get things into and out of the cell and have enough material inside to allow them to function properly, there's a happy medium for this surface area to volume ratio that makes them work the most efficient as they can. Right? So this is the phenomenon that keeps the size of the cells within a certain size frame. So prokaryotic cells, remember these are bacteria cells. They have no nucleus. They do have DNA, right? And that DNA, I'll put DNA there, is found in a specific area of the cell we're going to call the nucleoid region, right? And we're going to put a star by that because it's found only in prokaryotic cells, and so that's what those bolded ones are found only in prokaryotic cells. There are rounded globular structures in here called ribosomes. These are organelles that help produce proteins. It has a plasma membrane. We said all cells have that. Right? And then most or all bacterial cells will have a cell wall. It's a structure that helps them, gives them support, and helps protect them. All right? Now, bacterial cells also have a surface coat on top of that cell wall, and that coating is called the capsule. And that's a star by that. Okay? Found only in prokaryotic cells. Sometimes the capsule will have finger-like projections off of it. All right? And these finger-like projections are called pili. And pili is found only in prokaryotic cells. Okay? So those are the three things found only in prokaryotic cells. Bacterial cells might have a long structure that allows them to move through semi-fluid or fluid environments. This is called a flagellum. Several of them are called flagella. They're long whip-like structures. Most of the time they have 1 to 2 or 3. That's about as many as they normally have. All right? Flagellum. Okay. And there are eukaryotic cells that have flagella as well, right? Sperm cell is a good example of that, right? So those are prokaryotic cells and their structure. So the rest of chapter 4 deals with eukaryotic cells. Remember, they have a nucleus, and they have many organelles, and different types and different functions. And we're going to talk about those. So the first is a nucleus, right? So we're going to draw an animal cell here. And then inside the nucleus, the function of the nucleus is to contain or house the DNA, right? So we have DNA molecules in our nucleus, all right? And there are some things there, but the two most important terms for the nucleus will be chromosomes. And chromosomes are the DNA molecule, and then it might have some proteins attached to that DNA molecule to make it a chromosome, all right? All of the cell's DNA or chromosomes will be called the chromatin, all right? Okay. So one chromosome, and then all of them together are called chromatin or chromatin, all right? And they're all in the nucleus, right? In humans, we have 46 chromosomes. We have 46 DNA molecules, and therefore we have 46 chromosomes. Don't worry about that one. Okay, so there's a series of organelles in eukaryotic cells that are membrane sacs. All right. So we're going to move these out of the way. So we got our nucleus. And then there are structures right, that have these globs associated with them. Rounded structures. All right, with its lining. This is called the rough ER, endoplasmic reticulum, and ER is fine. So the rough ER, and the rough ER is a structure that, with the help of these little globular things called ribosomes, we talked about that over there, these produce proteins. There are also some tubular type of organs that don't have those ribosomes, and we call those smooth ER, and they make lipids. All right? Smooth and rough ERs. They do not have ribosomes. The next structure has three different functions, all right? And it's like a stack of pancakes, these sacks, all right? And what it does is it accepts these proteins and lipids from the ERs, it finalizes them, sorts them out, and then packages them so that they can be moved throughout the cell to do the work of the cell wherever they are needed. Okay? So the Golgi body or apparatus is sort of like a distribution center. The rough ER is more like industry. In animal cells, and put animal cell only, make sure you know this, these are little sacs called lysosomes. They have enzymes in them, and they digest cell waste. So there's some things in the cell that will be toxic to the cell if it's left alone. And so these lysosomes go around and get rid of, digest those molecules, and so they are like garbage men going around the city picking up cell waste. Transport vesicles, so we have these little proteins and lipids that are made in the ERs. They're in little sacs that move them around the cell, all right? They're like little transport trucks, and these are called transport vesicles. Vesicles, all right? So they're just little transporting membranes that carry the molecules around the cell. Also in the cell are these structures that are bean-shaped. I'm going to draw a larger one here. Kidney-shaped. Now these are called mitochondria. All right, mitochondria, mitochondrion. These do have a little bit of DNA associated with them, and sometimes DNA analysis uses that to help identify a person. All right. And this is the site of a process called cellular respiration. This is the way that the cell obtains chemical energy from glucose. All right. And so this is sort of like the the powerhouse of the cell, sometimes it's been called, all right? It's the site of cellular respiration. So lysosomes were found only in animal cells. In plant cells, they have some structures that are found only in those as well, okay? Has a nucleus and has mitochondria and all that kind of stuff too, right? But there's some other stuff that it has that the animals don't have. And they have these structures, they can be different shapes depending on the species of, but these are called chloroplasts, right? And the chloroplast has actually three layers, three membrane layers. And they have these discs. These discs are called thylakoids. Thylakoids contain chlorophyll. It's a pigment that allows plants to produce their own food. Okay, take solar energy and convert it into chemical energy of glucose. So thylakoids are these structures found in chloroplasts, and chloroplasts are the site of photosynthesis. That's where the plant cell makes their own food. They also have a large structure in the middle called a central vacuole. That structure has several functions. One, it is used to get rid of cell waste. Another thing is that it is a storage place for things like amino acids and sugars and things that the cell might need. And lastly, it regulates water balance. We'll find out that the plant cell has a wall around it, and so therefore it can't gain a lot of water. If it does, it will burst, all right? So the cells regulate how much water is in and out of the cell. And the plant cell uses this thing called a central vacuole to help with that water balance. In plants, they have this rigid structure called the cell wall. And I'm going to erase this part right here because I think we're finished showing that. All right, so you can read things better. But in the plant tissue layer, you have your cells, and these cell walls have little channels in them called plasmodesmata that help plant cells communicate with each other. They also provide support so a plant can stand erect. All right, the cells don't fall on top of each other, and it provides protection for the cell. In plants, it's made of cellulose, and the polysaccharide that makes the cell wall of fungi is called chitin. In animals, like plants, have these channels that allow them to speak together. In a tissue layer of animal cells, the animal cells come in contact with each other, right? And this contact is called a cell junction. And there are three types. The tight junction is just this physical contact that each cell has with its neighbor, right? And it doesn't allow things to pass through very, very readily, right? Then it has some elastic type of things associated with them, made up of proteins, called adhering junctions, and these allow the cells in a tissue layer to flex without being broken apart. without the tissue being broken apart. So you have tight and adhering, and then between each of these cells, there are little channels that allow the cells to talk to each other, and these are called gap junctions. Right? So you have tight, adhering, and gap. Finally, each cell has eukaryotic cells have these things called cytoskeleton. And cytoskeleton allows for support of the cell, but really kind of cool is that it provides pathways for those little transport vesicles to be moved around the cell. So they're like roads for the trucks, delivery trucks. All right. Some of the cytoskeleton material, though, can be used for other functions. So we said, like in a human sperm, it has a long whip-like structure called a flagellum or flagella. Right. These allow for movement through fluid membrane, fluid environment. Cilia, on the other hand, are short cytoskeletal material, and these allow for the organism to move very quickly around the cell by using these little short hair-like structures. They sort of do the wave, and they move around, and these are called cilia. Right? And then during cell division, there's some cytoskeletal material associated with that. There's little things called centrioles, and these are cytoskeletal materials that produce structures called spindle fiber, and we'll see what they're used for later, but they move the chromosomes around during cell division. So spindle fibers and centrioles. Right? So this is chapter 4. I know that was pretty quick, but you don't want me to sit up here and pontificate. You want me to get through it, and you can rewind this, and see it again if you missed something. But chapter 4, again, was mostly dealing with types of things within the cell.